I'm honored to be introducing our second keynote of the 2025 symposium, Dr. Sachin Patel. Dr. Patel obtained his BS in biological psychology from the University of California, Santa Barbara, then completed an MD PhD at the Medical College of Wisconsin with a PhD in pharmacology. He completed his clinical residency in adult psychiatry, then joined the faculty in the Department of Psychiatry and Behavioral Sciences at Vanderbilt Medical Center, where he held the position of Blakemore Professor of Psychiatry and Behavioral Sciences and the Director of the Division of General Psychiatry. He joined the Feinberg School of Medicine at Northwestern University as the Lizzie Gilman Professor and Chair of the Department of Psychiatry and Behavioral Sciences in January 2022. His clinical interests centered around the treatments of patients with substance use disorders and co-occurring mental illnesses in the inpatient and residential treatment. Dr. Patel's research is focused on understanding how endogenous cannabinoid neurotransmitters regulate neurobehavioral processes relevant to affective disorders and the pharmacology of cannabis constituents as it relates to pathophysiology and the treatment of psychiatric disorders. His research has been consistently published in a wide range of high-impact journals, and his research program has been continuously supported by federal, nonprofit, and industry sources. Dr. Patel has served as a content expert for the National Academy of Sciences, is a member of the American College of Neuropsychopharmacology, American Society for Clinical Investigating, and has served in National Society leadership roles throughout his career. Thank you very much for joining us today, Dr. Patel, and for sharing your expertise with us.
Great. Thank you, Ken, so much, and to all the organizers and societies and institutions for this meeting and for the kind invitation. So I'm going to go ahead and dive right in. This will let me advance. There we go. Perfect. I don't have any conflicts of interest to disclose. So I'll start with just a little bit about what I'm going to talk about. I'm not going to spend too much time on the first topic. I know there have been other speakers on this, but just a little bit of a background on cannabis pharmacology and epidemiology. And then three real meat part of the talk will focus on understanding and teaching a little bit about the physiology and function of cannabinoid signaling systems in the brain. And then moving into describing some of the most well-known sort of dark side adverse consequences of cannabis use, primarily as they pertain to psychiatric disorders, including psychotic disorders and anxiety disorders specifically. And then I'm going to end with a few slides about how our laboratory is investigating some of the underlying biological mechanisms related to adverse effects of cannabis using mouse model systems. And that'll just give you guys a little bit of a flavor of kind of the work that we're doing. So this is really a hybrid talk on sort of educating the audience on some of the adverse consequences of cannabis as they relate to mental health consequences, and then a little neurobiology at the end thrown in there to give people a flavor of kind of what we work on in my lab. All right, so probably doesn't need to be repeated for this audience, but just so we're on the same page, I group sort of cannabinoids into three big categories, and two of those large categories are shown on this first slide. Of course, there are natural cannabinoids that come from plants, like cannabis plants that are shown here. These plants produce a variety of molecules, two of which are quite important and likely are well known to this audience. Of course, THC, or tetrahydrocannabinol. This is the primary psychoactive component of cannabis. It binds to a specific cannabinoid G protein coupled receptor and exerts the vast majority of the physiological and subjective effects of cannabis and its intoxication. And when thinking about both adverse and potential therapeutic effects, the majority of those are really caused by THC, and we know that from a variety of human clinical psychopharmacology studies, as well as a lot of preclinical studies. Cannabidiol, or CBD, is also produced by plants in varying ratios. Generally, plants that produce high levels of THC will generally produce low levels of CBD because one is a precursor. In contrast, high levels of CBD in, for example, hemp, those plants lack the conversion enzymes to make THC, so they'll have very high levels of CBD but very low levels of THC. And there's been a lot of interest in CBD from a variety of perspectives on the therapeutic side. And some of you might know that a purified version of CBD named Epidiolex was approved about 3 or 4 years ago for the treatment of epilepsy in certain forms of intractable childhood epilepsy. So there's a lot of interest around CBD, certainly in the addiction realm. So those are the two most important molecules. And there are lots of other minor cannabinoids, and some of those are shown there. Synthetic cannabinoids are another large group. These are obviously, as their name implies, synthetically produced in laboratories, some of them by pharmaceutical companies, others by academic labs. And these are very useful tool compounds for biologists to understand and interrogate the cannabinoid signaling system. But they've also been used recreationally. And there was a time about 8 or 9 years ago, certainly when I was practicing in Nashville, at Vanderbilt, where we were seeing a fairly regular pace of patients presenting for psychiatric care after consumption of these very high potency synthetic cannabinoids. And they were marketed under a variety of names, including Spice and K2 and things like that. That seems to have died down, but there was a time where this was actually a pretty significant public health issue around the country. I'm not going to belabor the point, but people use cannabinoids in all sorts of various formulations that really were not even thought about 10, 15 years ago, including oral, topical, vaporizations, concentrations, and many, many different ways to consume cannabis. And of course, again, not going to belabor this, but it's really more accessible than it ever has been in our country with the geographic sort of distribution of recreational cannabis following what you might expect in terms of political liberalization, with the West Coast and East Coast showing many, many states with adult use recreational cannabis, Illinois also, and then no legal access in many parts of the Midwest and South. Another thing to mention is that, I talked a little bit about THC and CBD concentrations, and THC being the primary psychoactive component. That really is solidified by the fact that cannabis industry breeding of plants has resulted in larger and larger percentages of THC containing products. So plants now can have anywhere from 16 to 25% THC, whereas back in 1995, you can see that was in the 3 to 4% range. So the cultivation of these plants has proceeded in a way that supports higher levels of THC, presumably for the intoxicating effects that people are purchasing recreational cannabis for. Cannabis use is also rising. Many of you may have seen this slide before, but in really every age group, and not surprisingly, in terms of percentages, younger adults represent the bulk of people that use cannabis. But again, even in the older age groups, there's been an increase since probably 2016 in terms of the percentage using in past year. All right, so that's just sort of a little bit of a basic background in terms of setting the stage. And the take-home message here is that THC is really the primary psychoactive component. CBD may have some important bioactive properties, but doesn't have psychoactive properties per se, and so might be attractive for some therapeutics development. It's more accessible, higher potency in terms of THC content, and cannabis use is only going up really in every age group in our population, and that has paralleled, of course, the increasing accessibility due to decriminalization and frank legalization in many states. All right, so turning now to a little bit of biology around cannabinoid signaling and its physiology and function in the human body. There are two major receptor targets for THC, as well as synthetic cannabinoids, all cannabinoids really. And these are G protein coupled receptors named CB1 and CB2. The type 1 cannabinoid receptor is primarily regulated and expressed and regulating CNS and peripheral nervous system function, so brain and spinal cord. And regulates important processes, including appetite, motor control, sensory perception, emotion, cognition, memory. And the CB2 receptor, also a G protein coupled receptor, phylogenetically quite similar to CB1, but expressed primarily, not exclusively, in the peripheral tissues, including many listed here: adipose tissue, bone, gut, liver, pancreas, even in the skin, and heavily within immune cells in the periphery. And their function there is to suppress generally the proliferation of immune cells. And so that's sort of the general distinction between the type 1 and the type 2 receptor. If we focus in primarily on the type 1 receptor, as one might imagine, the CNS effects of THC in cannabis are most tightly linked to psychiatric adverse consequences. And here's just some nice PET imaging of the cannabinoid receptor here on the bottom. And in contrast, I've shown the mu-opioid receptor PET imaging study on the top. I think the first thing that you could see most obviously is that the cannabinoid receptor is really widely distributed throughout the brain, whereas the mu-opioid receptor has a highly preferential distribution within the basal ganglia and ventral striatal circuits involved in motivation and reward, whereas the cannabinoid receptor is highly expressed throughout the cortex, and you don't see that as much with the opioid receptor. And of course, the subjective effects of cannabis and opioids are quite different, and so this makes a lot of sense. But just to not belabor the point, but the CB1 receptor that you're seeing here is one of the most widely distributed G protein coupled receptors in the entire mammalian brain, and that might be related to sort of the breadth and pleiotropic effects of cannabinoids and cannabis in terms of affecting everything from cognition to coordination to emotional processing, memory, appetite, and so many different processes is likely underlied by its really wide distribution throughout the brain. All right, so I mentioned before that the type 1 receptors are primarily a CNS receptor or nervous system expressed receptor. This is a diagram from a recent review article we published, and I know it's complicated. Can people see the arrow here? Or I'm sure this is a webinar, so maybe people won't be able to tell me that or not. Perfect. So we've labeled the type 1 receptor here in blue. Its primary localization at central synapses is within the presynaptic axon terminal, and that's shown here. And when it's activated by either endogenous ligands or by exogenous THC from cannabis plants, the activity of that receptor serves to function to inhibit the release of neurotransmitter vesicles from these presynaptic terminals. And so in this case, if this is a glutamate-containing presynaptic terminal, when CB1 is activated, it inhibits the release of further glutamate. Also shown here are some synthetic pathways for endogenously produced cannabinoids. This is that third group that I haven't talked about yet, but these are molecules that our neurons and other cells in the body and brain make that activate the CB1 receptor in a very similar way to THC. So again, we have synthetic cannabinoids, natural cannabinoids coming from plants, and we have endogenous cannabinoids or endocannabinoids that are produced by cells within our bodies, and they all activate the same CB1 receptor. There's two of those. One of them is 2-arachidonoglycerol, and is produced in the postsynaptic compartment of neurons, especially within dendrites and dendritic spines. Anandamide or arachidonyl ethanolamine is another one, and that's predominantly thought to be actually produced within the presynaptic terminal. There's two other locations where cannabinoid receptors have been found. The first one is astrocytes. So these are supporting cells that play important roles in metabolic regulation of neurons and also some signaling processes. And the type 1 receptor is expressed within these astrocytes and plays a unique role here in liberating gliotransmitters or glial-derived neurotransmitter molecules through increases in calcium within astrocytes. And then the CB2 receptor has been found within the brain, but specifically located to microglial cells. So these are immune cells of the brain, which makes sense, as I mentioned before, that the CB2 receptor is really an immune modulating cannabinoid receptor. And in the brain, the microglial cells are the primary immune cell. And so when the CB2 receptor is activated either by THC or by these endogenous molecules, 2-arachidonoglycerol or anandamide, the function of these CB2 receptors is to reduce cytokine and other inflammatory mediator release from these microglial cells. And in that way, cannabis and endocannabinoids play an important role in reducing neuroinflammation and inflammatory processes associated with brain injury and trauma and other types of neurodegenerative processes. So I know that was a lot, but that's kind of a whirlwind of what we know about what cannabinoid receptors do at synapses, as well as non-neuronal cells such as astrocytes and immune cells in the brain, including microglia. Once that cannabinoid receptor is active, there's a variety of intracellular processes that are triggered. And in the short term, these GPCRs can trigger reductions in neural excitability and reductions in neurotransmitter release, as I mentioned in the previous slide. There are also longer-term effects through cyclic AMP and protein kinase A-mediated effects on gene transcription and long-term changes in synaptic strength that can occur through genetic mechanisms. So the GPCR, CB1 GPCR, like many others, can signal through both short-term and long-term timescales and can interact with the genome to actually affect transcription and long-term change long-term synaptic strength through genetic mechanisms. All right. Again, so the take home here is that THC and other cannabinoids act via two primary receptors, the CB1 and the CB2. CB1 is really widely distributed in the brain and spinal cord. Inhibition of neurotransmitter release is a primary mechanism of action of CB1 receptors, and that occurs through both modulation of kinases and ion channels located within that presynaptic terminal and on microglia and astrocytes. Okay, so now moving on to some of the things that I've been interested in quite a while, both from a clinical perspective as well as a biological perspective, is really understanding some of these sort of negative adverse consequences of cannabis use as they relate to mental health conditions. And I'm going to talk about three of those. And the first one is cannabis use disorder. The other one is effects of cannabis on psychosis, including psychotic disorders, schizophrenia, schizoaffective disorder. And the third one is really anxiety and trauma-related disorders. Again, probably not a surprise to the audience here, but cannabis use disorder is going up, and this is a nice diagram showing sort of the pyramid of cannabis use, including low sporadic experimental use leading to increasing hazardous cannabis use, and then ultimately cannabis use despite significant adverse consequences resulting in criteria for cannabis use disorder according to the DSM-5. And this is an interesting concept of trying to understand medical versus non-medical cannabis use motives, and if there's any differential contribution of medical versus recreational cannabis to developing a use disorder. And not surprisingly, non-medical cannabis use was significantly more likely to result in more severe cannabis use disorder in terms of symptoms, for example, than medical cannabis use. And both use of both medical and non-medical use results in similar proportions as non-medical use alone. So this is, again, not surprising that non-medical or recreational use of cannabis is associated with a higher likelihood of developing a use disorder, as well as developing more moderate to severe use disorder in terms of number of symptoms experienced. Some risk factors, again, these are not necessarily specific to cannabis, but have been shown to increase risk for cannabis use disorder, initiation at younger age, male demographic, comorbidity of tobacco, cigarette use, and of course, unsurprisingly, greater frequency of use results in a higher likelihood of developing a use disorder. And again, not unique. Many of these risk factors are the same for tobacco and other substance uses. So nothing necessarily unique to cannabis, but data has shown that these are risk factors that also affect the development of cannabis use disorder and confer risk to the development of cannabis use disorder. Cannabis withdrawal, this was a new diagnosis that was added to DSM-5 about 10 years ago. A little over 10 years ago. And this was some of the seminal work done by Meg Haney and her group at Columbia, where heavy persistent cannabis users are brought into a very controlled setting and put into a withdrawal state. Her and her group were able to show measurable increases in things like irritability, reductions in sleep, increases in being awoken often at night, for example, reductions in sleep efficiency, as well as transient reductions in caloric intake and appetite. And those are the things that define the cannabis withdrawal syndrome in the DSM. So many of these things are sort of the opposite of what one experiences during intoxication, which is true for all drugs, of course, that the withdrawal syndrome is sort of the mirror image of the intoxication syndrome. And so with cannabis, of course, people use cannabis to help them sleep, to increase appetite, and to reduce anxiety or for its calming effects. And of course, the withdrawal phenotype then is characterized by the opposite of all of those: increases in irritability, poor sleep, and decreased appetite, and decreased caloric intake. These effects are really only seen in very heavy chronic users. It's unlikely that this manifests in recreational or sporadic cannabis use. And like many, most, if not all other drugs at this point, it is our understanding that the initiating effects, biological effects that trigger the sort of trajectory towards a use disorder involve activation of brain reward systems. I've shown this well-known diagram here showing the localization of CB1 receptors, primarily on these inhibitory neurons within the ventral tegmental area that contain dopamine neurons that project to the reward centers of the brain, including the ventral striatum or nucleus accumbens. And when this cannabinoid receptor is activated, it prevents GABA release. GABA is inhibitory, which then results in a disinhibition of these dopamine neurons. And it's thought that at least the positive reinforcing effects of initial or sporadic cannabis use may be mediated through this mechanism. Of course, there's a longer-term trajectory that leads to a cannabis use disorder. I'm happy to talk about that later. But the localization physiology of the receptor supports a mechanism by which dopamine neurons get disinhibited during the intoxication state and activation of this receptor on these inhibitory cells. So the second area I wanted to talk about was the well-known effects that cannabis has on psychosis. And this is a conceptualization that I borrowed from Dr. Cyril D'Souza at Yale that's done a lot of the seminal work in this area. And he conceptualizes the effects of cannabis on psychosis and psychotic disorders into three sort of broad categories that really are sequential to some degree. And so I'll tell you a little bit about what I mean by that. And the first is that acute cannabis intoxication can, under some circumstances, lead to a psychosis-like state and can mimic the effects of psychotic disorders and generate psychotic symptoms in some patients. But in many cases, and in most cases, this dissipates once the acute intoxication is over. There's a second interaction where there's a prolonged period of psychosis or psychotic symptoms that are present even after that acute intoxication phase has dissipated. And this is sort of a persistent psychosis, psychotic-like state that occurs in patients using cannabis. And then the third conceptualization is understanding how cannabis use, especially during development, represents a risk factor for the overdevelopment of psychotic disorders later in life, like schizophrenia and schizoaffective disorder, for example. So I'll talk about each one of these in a little bit more detail. So this is some recent data looking at the prevalence of acute cannabis-induced psychosis. So this is during the intoxication. And again, rates are relatively low if we look at lifetime or last year, and this is of people using cannabis, about 0.5%. So, 1 in 200 at most, although this might be underrepresented because this is taken from people that actually seek medical care for the condition. It's likely there are many, many people experiencing these symptoms that don't necessarily seek medical care. So this might be an underestimate, but, I would say anywhere from 1 in 100 to 1 in 200 based on this data. And if you look at a little bit of the phenomenology of what people experience and what leads to this state, you can see here that generally people are using high potency cannabis here in the dark green, which makes sense. More THC, more likelihood of eliciting side effects. Amount of cannabis. So, it's really not the amount, it's more the potency because most people are using 1 gram or less. And there doesn't really seem to be a lot of comorbid drug use that's interacting to produce this state. For example, if you look at alcohol, most people that develop cannabis-induced psychosis are not drinking alcohol. Some of them are, but it's usually a small amount, between 1 to 5 drinks. And then if you look at other comorbid drug use, almost 50% of the people are using no other drugs. And the second biggest group here is alcohol, and things that you'd imagine that might contribute to the psychotic state, such as amphetamine and cocaine, actually are a small group. So it seems like cannabis, high potency cannabis, really not in the context of comorbid or co-use, really has the ability to produce these psychosis-like states in again a relatively small group of patients. This is some seminal data by Cyril D'Souza, who I mentioned earlier, working at Yale. This is a human experimental study where he actually gave intravenous THC or a placebo to healthy volunteers and measured both positive and negative psychotic symptoms. So positive symptoms include things like delusional thinking, hallucinations, perceptual changes. Negative symptoms include things like cognitive symptoms, emotional blunting, and disorders of thinking, such as thought blocking, for example, an inability to organize one's thoughts in a clear and logical way. And you can see here that both 2.5 and 5 milligrams of THC caused a transient increase in the symptoms. Again, these are healthy volunteers with no psychopathology. So even in seemingly patients that do not have a psychotic disorder, you can get these transient psychotic-like symptoms. And again, at relatively high doses, and yet using an IV route, which is obviously not what people do. But the point here is that THC does have the ability to produce transient psychosis, which parallels the epidemiological data that I showed in the previous slide. In terms of the second group, which is sort of a persisting psychosis, there's actually not that much hard data on this. I'll just give you my anecdote that when we were seeing people specifically using synthetic cannabinoids about 10 years ago in Nashville, when they were being sold over the counter and on the internet, we would see probably one patient every week or two that would be admitted to the hospital with a persisting psychotic syndrome after use of these highly potent synthetic cannabinoids. And some of these are up to 100 times more potent than THC. And the problem is people don't know how much they're taking or what they're taking. And we would see these symptoms lasting for up to a couple of weeks. And so this was clearly past the acute intoxication phase, but there appeared to be this sort of persistent syndrome that lasted many days and sometimes even a few weeks. And there have been some case series and case reports of these persisting type disorders that last many, many days after cannabis use. And then I just want to spend a few slides talking about the third group, because really there's a lot of data suggesting that cannabis use, especially during adolescence, represents a risk factor for increasing the odds of developing psychotic disorders, which include schizophrenia and schizoaffective disorder later on in adulthood. And both of these show the odds ratio for developing schizophrenia by either a cohort or cross-sectional studies that have been conducted. And here the meta-analysis showing risk anywhere from 3 up to 4.5 over non-cannabis users. And also using outcome measures as symptoms or an overdiagnosis. Again, the risk is up to 4-fold higher for people that use cannabis than that did not, in terms of their risk for developing psychosis. This was a nice study showing that looking at studies that provided some sort of dose-response relationship of how much cannabis people used and their likelihood of developing a psychotic disorder later in life. And you can see that all the studies that did this showed that the more people were using, the higher their risk, suggesting some sort of dose-response relationship between cannabis use and a subsequent development of a psychotic disorder. And then lastly, this is an interesting study showing that both males and females that use cannabis develop psychotic symptoms at an earlier age, suggesting that cannabis use during young adulthood and adolescence might accelerate the onset of a psychotic disorder in people that may have genetic or other susceptibility to going on to develop one in and of itself. So acceleration of the onset of the disorder also might be a way cannabis interacts with people that have a genetic liability for schizophrenia. And again, interestingly, the accelerated onset in age is related to the potency. So people that are using high potency skunk type cannabis every day show the youngest age of onset of a psychotic disorder. Again, these are all people that were going to likely develop psychosis, but the people that used cannabis seem to develop that at a much earlier age. So this more rapid acceleration of the disease pathology in people using cannabis. All right, so we've got about 15 minutes or so left. So the last part of this talk is really going to focus on cannabis and anxiety and a little bit about the clinical data, and then going into some of the neural data that the preclinical approaches we're taking to try and understand this interaction between cannabis and anxiety. And similar to psychosis, I've sort of conceptualized the interaction between cannabis and anxiety disorders into two categories. And the first is, again, the acute intoxication phase. It's well known that cannabis can affect anxiety symptoms during acute intoxication. And then the second, again, is cannabis use and its risk for the development of subsequent anxiety disorders. So we'll take the first one first. But before we start with that, it's important to know that when you look at reasons why people use cannabis, both recreation and medical cannabis, the number one reason in terms of psychiatric indications is really anxiety. So people use cannabis to cope with anxiety and stress. And then you can see that here listed as the first thing when you simply ask people that use medical cannabis, why do you do it? Pain is number one, but if you look at the non-somatic and go down to the mental health conditions, anxiety is uniformly number one. And this is another way of looking at that in terms of the types of patients that dispensaries are marketing to. It's anxiety disorders is almost the number one thing, and then pain is number two. So of people that use cannabis chronically, the vast majority of them, especially for medical cannabis, will cite coping with anxiety as the reason that they use it. Paradoxically, we also know that cannabis-induced anxiety is one of the number one reasons why people stop using cannabis. So it's this very interesting dichotomy in terms of chronic users citing relief of anxiety, whereas cannabis also being known the number one psychiatric adverse consequence of cannabis use. And you can see that here, where after headaches and dizziness, anxiety is the number one adverse effect noted. And again, here, this sort of self-report survey done by LaFrance here at Washington State University. After coughing, anxiety is the number one in terms of prevalence adverse effect of cannabis. And another study showed that of 1,100 people that were evaluated for acute cannabis toxicity in the ED, almost 20% of them had a chief complaint of anxiety. And so our conceptualizations currently are that low doses of cannabis under environmentally sort of calming situations is likely to have some benefit or perceived benefit in terms of reducing anxiety states and having a calming effect and a stress reducing effect. However, as the dose gets higher and the environment gets more stressful, there can be this sort of inverted U shape where now people start reporting increasing symptoms of anxiety and overt panic attacks can actually occur. And then at very high levels, you can start getting psychotic symptoms, as I talked about in the previous several slides. So there's a real sweet spot in terms of dosing of this particular drug where lower the better, but once people sort of pass a tipping point, they can start getting a lot of adverse effects. And so then what about the second group, the category of cannabis use and then subsequent risk for anxiety disorders? And there's been some really nice, very convincing epidemiological data coming out recently. And this is one study that came out about a year ago showing that people that had an emergency room visit for some adverse effect of cannabis use were much more likely, up to fourfold more likely to present to either a hospital or an outpatient clinic in the subsequent 5 years for some sort of anxiety disorder. And so these are the curves here showing the general population of people over 5 years that would seek treatment for an anxiety disorder. And the blue line here shows people seeking treatment for an anxiety disorder that had some point in the previous 5 years presented to the emergency room with an adverse complaint of cannabis adverse effect. And so you can see here in the color coding, this is down by age group, that the highest risk or the highest likelihood of someone showing a diagnosis for an anxiety disorder after experiencing adverse effects of cannabis was predominant in the sort of younger age group, 18, 20, 24. Then there's a little bit of a lull here as the age group gets higher, and the same is true for males, both males and females. It generally seems to be a little bit higher for females. And the secondary outcome is, again, similar. It's a hospitalization for an anxiety disorder. But again, that young age group, 18 up to about 25 to 40 is where the bulk of the signal is coming from. It's not really coming from older adults. So there does seem to be some evidence that cannabis use resulting in pretty significant adverse effects, enough that you had to go to the emergency room, has some predictive value showing that there's an increase in the odds of developing some sort of anxiety disorder within the subsequent 5 years. So the data here is not quite as strong as for psychotic disorders, but it does seem to be emerging that there may be a link between cannabis use to the point where it's causing an adverse effect and subsequent development of anxiety disorders. So I'm not going to reiterate that because I want to spend the next 5 minutes or so really talking about some of the ways that we have been interested in trying to understand the biology of this mechanism. So this is a slide that I showed before where there seems to be this, we have a hypothesis, and it's supported by a lot of data, that as the dose of cannabis goes up and the environment becomes more stressful, people will tend to have more adverse effects, including anxiety-like symptoms and panic attacks, and then potentially even psychotic-like symptoms. And so this was a study we did almost 20 years ago in mice. And the bottom one is a study in rats done by a colleague of mine at University of British Columbia, where we gave animals THC or a variety of different synthetic high-potency synthetic cannabinoids under 2 conditions. One would just be a normal condition in which they're sitting in their home cage, and another condition is when they were exposed to stress. And what you're seeing here is a brain region known as the central nucleus of the amygdala. All of these light brown dots are neurons, and then the dark brown or the black dots here are neurons that show increases in activity. And the take home here is that when we combine a high dose of cannabinoids, whether it's THC or a synthetic cannabinoid, with stress, we see a big increase in the neural activity within this central amygdala. And this is a nucleus that's really critically linked to generating fear-like states in humans and primates, as well as in mice and rodents. And again, here you can see another example of the central amygdala showing a big increase in neural activity in animals that were given a synthetic cannabinoid, but also combined with stress. When you give the cannabinoids without stress, you really don't see much activity in this area. So we've known this, again, for about 20 years, that the central amygdala is probably a key hub where cannabinoids interact with these particular neurons to potentially generate anxiety-like responses. And so in the intervening 20 years or so, a lot of research, primarily in mice and rats, has taught us a lot about how this central amygdala regulates these defensive or fear-like responses. And the take-home sort of you see here on the right, there have been multiple studies that have identified different types of neurons within the central amygdala. And it's suggested that these different subtypes of neurons will generate different types of defensive responses. For example, this PKC expressing neuron seems to be important when investigating potential threat. So examination of odors or stimuli in the environment that might have some negative adverse consequence. These somatostatin cells, or SOM or SOM neurons, seem to be important for generating like freezing responses when a threat in the environment is detected. And these corticotropin releasing factor, or CRF cells, might be important for escape or ballistic sort of flight-like responses to escape from imminent threat. So different neurons may have different roles in generating different types of these fear responses. And in mice, these are the ones that they express. They express freezing behavior, they express jumping behavior, escape behavior, and investigative behavior. And this is where a lot of the research in this area has been done. So we really focused on trying to understand one type of neuron, this somatostatin cell, this SOM neuron, and how cannabinoids might be interacting with stress to affect the activity of these cells, because we know in mice, when you give animals high doses of THC and other cannabinoids, they show a lot of freezing behavior, especially to stress. And so we thought that this would be important to understand, because it might give us some insight into how THC produces, one, the adverse effects, including anxiety and panic reactions in humans, and then more long-term might give us some insight into how early cannabis use might present or might lead to sort of a longer term risk for developing anxiety disorders. The second part is really something we haven't delved into, but a lot of the work I'm going to show you might allow us to do that in the future. So the first thing that we wanted to do was try and test whether these somatostatin neurons that are really responsible for generating these freezing responses to stress are actually active more when the animals are given cannabinoids. And so this is an experiment that we did in mice. We were able to access these somatostatin neurons in the central nucleus of the amygdala with a little microscope that we placed down into the mouse brain, and that allows the animals to walk around in their home cage, and then we can give them different drugs and look at the activity of these cells by how much calcium is coming into the neurons, and that's a surrogate marker for how active those cells are. And so this is just a picture of kind of what it looks like when we put the microscope down into the brain, and when we extract a movie, these are kind of the neurons that we can see. And then here are some of the actual data that we would get from a particular cell. If we outline that particular cell here, for example, we can see these calcium traces, and every little upward deflection here represents an increase in the activity of that particular cell. And if we plot this as a heat map now, where red is basically more active and blue is less active, we can see that after cannabinoid administration, in this case, a synthetic cannabinoid called CP, it's very structurally similar to THC, we can see that there's a lot more activity compared to the vehicle group here. So yes, indeed, we're seeing an increase in this particular subtype of neuron that we know is critical for generating these fear-like responses or freezing responses in mice when we give the animal a cannabinoid. So at least we know that cannabinoids are increasing the activity of these particular cells in mice. And then we did a lot of computational work to try and understand how the coordinated activity of these cells might be changing. And so this is just one example of what we found that was quite interesting. So this is a diagram of, in one particular mouse, all of the neurons that we were recording from. And so we can see it in a circle here going from 1 to 58. So there are 58 cells in this particular animal. And this is the pre-drug condition. And the lines here in the red represent how connected these cells are in terms of their correlated activity. So if two cells fire together all the time, they would be highly correlated, and you would see a very strong line sort of between those two. And what you can see here is two things. One is there's a lot of correlation between cells, but there also seems to be a high degree of correlation between cells that are close together. And these aren't necessarily cells that are close together anatomically, but these are cells that represent a community where their activity is more intercorrelated and synchronous together. So we sort of place them in a way where those ensembles are closer together here in numbers, which is why you're seeing a lot of activity together. When you give the animals a cannabinoid, one thing you notice is that that's magnified dramatically, where there's a lot more activity within these ensembles as opposed to between them. So the between communication is reduced and the within communication goes up. So what we think this means is that these subgroups of somatostatin neurons are much more synchronous, and that's why they're more highly correlated. So now we're beginning to see how THC can actually affect the neural dynamics of these particular cell populations in a way that might support the generation of these defensive responses. And that's something we're investigating as to how this hypercorrelated activity within small subgroups might then increase freezing responses or fear responses in these animals. So I know I'm running out of time here. I'm going to skip this and just tell you about maybe this slide and then end there. What we really wanted to know was how cannabinoids interact with stress. And so in this particular experiment, what we now did was while we were imaging these neurons, we put a predator odor, this is a derivative of a fox odor, in the cage. And when that odor is there, the animals will show some approach behavior, they'll run away from the odor, and then they'll start freezing when they figure out it's not something that they want to be around. And we can record the activity as the animals approach the odor, run away, and as they freeze. And we were able to see actually very interesting things that in the animals as they approach the odor, you can see a ramping of activity under the vehicle condition, but after the drug treatment, that seems to be dramatically magnified. And in addition, there are cells that show a reduction in activity as the animals approach, and that is also magnified. So what we think is going on is that THC is really upscaling the responsivity of these cells to threats. So the cells that normally go up more. The cells that normally are inhibited more. And so there's just a magnified neural representation of these threats in the environment, which then presumably would trigger stronger freezing responses. And that's something we're looking at. So I'm going to stop there because I want to make sure we've got time for questions. But that's just an example of some of the approaches we're taking to understand what are the cell types within the brain that cannabinoids affect, and then how cannabinoids are changing communications within these cell types and between cell types. How are they changing the representation of things in the environment, like threatening odors? And then how could we understand how cannabinoids then might be really subjectively changing the fear state, or at least in mice, the behavioral responses to fear that might give us some insight as to what's going on in humans. And hopefully we could use a model like this to, for example, treat animals with cannabinoids like THC or synthetic cannabinoids, for example, early in life, and then test them when they're adults and see if they're more likely to have anxiety-like phenotypes in response to stress. And does that relate to long-term changes in the activity of neurons like these somatostatin cells in the central amygdala? And I think all of that might provide us insights and give us some biological mechanistic basis from understanding how cannabis exposure, especially during development, might set people up for predisposing them to developing long-term psychopathology in the form of anxiety disorders or things like that. So I'm going to stop here and just want to thank many of the folks in the lab that worked on these, as well as some of the funding sources for the experimental data that I showed you. And I'm happy to take questions.